Hi there, and welcome to this SpudSmart webinar on fighting common scab. My name is Carrie Belanger, and I'm the editor of SpudSmart Magazine and SpudSmart.com, and today I'll be acting as your moderator. First, I'd like to thank our sponsors, McCain Foods and BASF, for their support. And this webinar is being recorded and will be made available on SpudSmart.com following the event. For audio quality purposes, all microphones have been muted with the exception of the speakers, but we do welcome your input. Today, we have three guest speakers. George Lazarovits is the Director of Research at A&L Biologicals. Claudia Goyer is a molecular bacteriologist for Agriculture and Agri-Food Canada. And Yahoo Wang is a professor and potato researcher. Robert Coffin, unfortunately, was unable to join us today. Some of the items on our agenda today include scab-resistant varieties, factors driving common scab severity in soils, improving resistance of existing potato varieties using somoclonal variation, biocides used to control common scab in potatoes, and field trial results, among many other topics. Please type your questions or your comments into the chat box at any time during the webinar, also indicating to whom the question is directed. At the end of the complete slide presentation, there will be an opportunity for each guest speaker to answer the questions posed in the chat box. Our first guest today is Dr. George Lazarovits. George is the Director of Research at A&L Biologicals, based in London, Ontario, which implements molecular technologies for the detection and diagnosis of microorganisms that impact farm production. Please take it away, George.
Thank you, Carrie. I hope you can all hear me. Thank you for inviting me to give this webinar. One of the beauties about giving a presentation like this, they tell me that I can sit in my lab office naked and you won't even know it. So I would like to just give you a little bit overhead about some of the work we have done on impacting potato scab disease. This work was sponsored by the potato growers of PEI in Ontario and by Horticulture Australia Limited over a number of years. And so what I'll try to do is give you a little bit of impacts on economic losses, some key factors that we do on diagnosis, potential sources of inoculum, and role of soil chemistry and fertility. All of them are complicated issues, but I'll try to make them as simple as I can. So this bacteria, when I was a graduate student, was actually considered a fungus because, as you can see, it grows almost like a mycelial type of component, but it was very early recognized as a bacteria, a gram-positive bacteria. And now we have come to learn that there's a whole bunch of species, and we're not quite sure yet which are the most prevalent, but I'll get to that later on. So when you plate, we developed a selective media for growing these bacteria. And unfortunately, when you plate on this media, you can't identify which ones are the virulent species. Although generally speaking, the ones that make this brown melanin pigment that you see with my green arrows tend to be often the parasitic types. Now, the only way you can tell which ones are parasitic is they make a toxin called thaxtomin, which was isolated in New Brunswick by Agriculture Canada. And this is what the toxin looks as a yellow band on a thin layer plate. And what this toxin does, it's a very unique compound. It's one of the only ones recognized to inhibit specifically cellulose biosynthesis in plants. And I believe a patent has been applied for as a herbicide. I'm not sure it will make a great herbicide, but in any event, understanding the toxin production by these organisms is the key to way a key way to quantitatively determine their presence. Now, in my career, I have been frustrated often by lack of resources to do the research on scab. It's not exactly on the top list of any government agencies for funding. So at one point, we got so frustrated in having sat down with a number of growers, and they were showing me their bills of lading and how much of their yield was deducted by the companies taking the produce based on scab prevalence in the load, that we actually surveyed about 1,000 farmers, and we got 233 responses. As you can see, that we estimated in 2005, and this was done by with my compatriot Jackie Hill, who was a terrific statistician, that we're losing about somewhere in the ballpark of $20 million a year at that time. Now, it may be much higher. And of that, of course, at that time, Quebec was taking the largest brunt, followed by PEI, then New Brunswick. And as you can see, Ontario was a lot less, and the prairies at that time were also very marginal. But if you look globally, this could represent billions of dollars losses, where China and the United States, and I just came back from Pakistan, all report extremely severe scab issues. So just recently, we have tested with Eugenia Banks 50 farms for the types of scabies or the types of streptomyces causing disease in the fields. And like published by Leslie Warner back when, we found that all of the scabies isolates that she reported were present in Ontario, and I believe the subsequent speakers have similarly shown that it's also present in New Brunswick and Quebec and PI. So we do not know very much about the physiological and ecological behavior of these organisms. We do suspect that they have very different requirements for establishing soils, and they may have very different virulence components. But again, this all needs to be studied at some point. Okay, so one of the things that we have developed in A&L, and I think it's probably available at other labs, is a quantitative measurement of the bacteria. So what you see in this particular graph is DNA isolated from 10,000 bacteria, 1,000 bacteria, 100, 10, and 1 bacterium. And what we are targeting in this type of PCR, this is polymerase chain reaction, which is a molecular technology, is the gene related to the production of the toxin. So only this bacteria, of course, we don't know what species because they all have the gene, only this will show up in our analysis. And what you see here is that the earlier you detect the presence of the bacteria, that means the more concentrated the bacteria are. And by drawing a line through this, one can develop a standard curve. And if you send us a sample, we can tell you exactly that you have 565 bacteria per gram of soil or whatever. But this technology is very inexpensive. It's very rapid. As you can see, it takes about an hour and 15 minutes. This represents the number of cycles at the bottom that you get for each time this replication occurs. So if anyone's interested, we can certainly provide this service to them. Very often, I will get calls from growers about an issue that comes up in the spring. They'll call me and they'll say, George, I just got a big load of seed in from this and this location, and it's full of scab. And if I plant it, will I have scab in my field? Will I contaminate it? And I usually reply saying, can you get clean seed? And they say, no, I can't. Well, I will then say, all right, plant what you got, because we don't know if you'll get scab. And I'll explain why. But we did do a test on a number of seed lots over the years. In 2004, I should say, we tested 41 lots of seed from different locations. And what we did was we looked at it, whether it had visible scab or no scab visible, and we took samples and measured the populations of the scab. And as you can see, there were 11 lots that had in the 10 million scab organisms per unit of area that we sampled. 19 had no disease symptoms, but the scab was present on the contamination on the surface of the seed tubers. And 11 were apparently clean, but we don't know because we didn't sample the entire surface. This represents 3 tubers from each seed lot that was analyzed. So you're looking at about 70% were contaminated versus, and we did it again in, excuse me, in 2006 with very much along, this time we got some samples from Alberta also. And we are finding that 15 lots with visible scab, 14 lots with pathogen on the surface, and 8 were clean. So very similar results in both years. And one of the interesting things is that whereas you may get scab, surface covered scab that you can see the lesions, you will also find that tubers will have the pathogen on them, but only on the surface soil layer or whatever. And that doesn't mean that you're not transferring that pathogen into the next crop. Well, the question then became is, do infected seeds or infested seeds transfer the bacteria to the soil? So what we did was we took some pasteurized soil and we placed it into these round pipes that you see here, which were then buried in a microplot. And then we put into them tubers that had visible scab lesions, tubers that we knew had surface scab organisms on them, the Streptomyces bacteria, but there was no visible symptoms. And then we took some tubers and we dipped them into alcohol to make sure that they were clean. And then we grew we went, and as you can see in the bottom panel, we sampled all of the regions around the tubers to see where the bacteria were coming out of the tubers from. And what you see here is you're seeing that yellow grid around the mother tuber, and you can see that at the bottom of it, we're getting 10,000 to 100,000 bacteria being released from that mother tuber into the soil, but there's very little bacteria above the soil layer. So there is no doubt that the bacteria are emanating from the infested tuber into the soil. In this case, the soil was pasteurized, so they may have had an easier time establishing. And the results, as far as what the crop looked like from these tubers, you can see that where we surface sterilized, we had hardly any scab in the tubers that were formed from the mother tubers. Where we had the surface bacteria present but no visible lesions, 61% of the tubers had scab, a visible scab incidence, and where we didn't surface sterilize and put scabby tubers, it was 88% of the tubers were showing visible symptoms. So there is a good chance that when you plant infested seed, you will get some transmission. Now, at the same time, I have had growers that told me that you always use scabby seed and I have no scab in my field. And those are the fields that are probably worth a lot of money because they may be the ones that suppress the spread of the organism. And hopefully one day somebody will find this and find out why that the bacteria does not spread in some fields where it does in others. So the recommendations for this says, if we have now developed rapid and inexpensive means of seed disinfestation, or we need to do that. We tested 70% alcohol spray, and it worked well, but we think it's too expensive. Although a couple growers in PI said they tested it and they planted these tubers in an infested field where they had a lot of scab, and they reduced the incidence by 50%. So there's some hope for this, but we need to do some economic analysis, and we need to find some other things than perhaps alcohol to do this. Acetic acid may be an option. It's used in Europe, and there are other potent biocides such as that may be available for seed disinfestation. The other component that was always controversial, particularly in A&L and with the grower recommendations as to fertility, is what makes scab worse as far as fertility goes in some soils and not others. So we sampled a whole bunch of plants from PEI in Ontario. We dug up 5 consecutive plants from 3 locations in fields. And when we removed the tubers and we took the soil right from the surface of the tubers and analyzed them for bacteria and the chemistry of the soils attached to the tubers. And then we rated the scab severity, and we also related the total chemistry using various analytical methods to the factors that may be contributing to scab. And this paper was published in Phytopathology in volume 97. If anyone's interested, I can send it along to them. Okay, so I will go through this somewhat because the results are rather interesting. Let's take the PEI soils, where you can, so the scab index of 2 or higher means that these tubers are not marketable. Anything less than 2 here, we could potentially market. Now, in PEI, we didn't find too many soils where there was low scab. We found a lot that had high scab. What was interesting that where you find the pH was below 5, we had low scab. Where we had 6.1 or higher, it was high scab. Now, if you look at Ontario with the same results, their pHs were 77.3 with low scab and 70. Now, you notice that 161 samples had very low scab and only 40 had high scab. But the pHs were almost identical. So there's no correlation in this soil between pH and scab incidence, whereas there is in PEI. Organic matter I won't deal with, but potassium, percent potassium in the soils was significant in both PEI and in Alliston. And what you'll see is that in the low scab in PEI, we had a potassium was at 5.5, whereas in PEI it was 8.8 for the high scab. And again, in Ontario, this was very similar, 5.8 versus 7.8. So somehow potassium is making the disease worse. Magnesium, on the other hand, probably makes the disease less severe, and I'll explain that later on. But in PEI, we found 6.3 and 8.2. But if you notice in Ontario, it was not significant, but the levels were almost 3 times higher in the magnesium level, 17.9 versus 15.1. Again, the lower level with magnesium had less disease. Calcium was only significant in PEI, whereas in Ontario was not, even though, again, the calcium levels were double that of PEI. So it's hard to understand why this was correlated. And it may be something that's not relevant. Sodium was not a major factor. There was some marginal effect. Manganese was interesting in that in PEI was only marginally related, but it was both low, 23.7 and 29.5. But in Ontario, it was almost triple where there was no scab and almost double where there was scab. So manganese may have some kind of role. But of all the factors we found, the most significant was the K to mag ratio. Wherever we had levels of 0.4 or less between the K to magnesium ratio, we found very low disease. And this was proven in many, many soils, but not all the soils. And in Alliston, you can see that 0.57 was already significant higher. In PEI, we never found a ratio anywhere near 0.4. It was always above 1. And perhaps that's why we're getting such high incidences. This may be something that needs to be followed up by further study down the line. So my recommendations are that high potassium in the absence of magnesium seems to increase scab severity. Soils with K to mag ratios around 0.3 and 0.4 invariably had the lowest disease scab severity. It doesn't mean they had no scab. They were just lower than anywhere else where the potassium-magnesium ratios were above 0.4 or below 0.3. So this seems to be a window of low levels of disease. Soils with higher levels of manganese had lower scab severity overall. And pH does not appear to play an important role in all soils. So what we need to know is a lot more about the chemical and biological properties of the soil that impact the various species of scab-inducing bacteria. And I think as a research priority, we should be looking at to see how the various species of scab organisms function under these different recommendations of K or mag or manganese. And I'll leave it at that, and thank you very much. Oh, one last slide. Sorry. The salvations for this may be that if we can develop resistant varieties, and there are already some varieties that have some degree of resistance, but we're not sure if they're resistant to all the strains of the pathogen, as we don't know what are the predominant strains in most soils. And secondly, we still have many reports about scab suppressive soils where the pathogen is present, but disease does not occur. And why this happens would really, really make everybody very happy. Thank you very much.
Thank you so much, George, for your presentation. Our next guest is Dr. Claudia Goyer. Claudia is a molecular bacteriologist for Agriculture and Agri-Food Canada. In addition to potatoes and common scab, her areas of expertise include biopesticides, resistance, microbial ecology, the nitrogen cycle, and greenhouse gas. Please take it away, Claudia.
Thank you, Carrie. Thank you for the invite. It's my pleasure to subject that is very dear to me. George has done an excellent job at giving like the background of common scab and the pathogens. So I'm not going to repeat that. I'm going to jump in just in the meat, in the thick of it. So as George have kind of indicated, it's very difficult to control common scab. So there's no product that you can use and all of your problems are going to go away. There's some success in some agricultural practices. If you look in the literature, like rotation, application of manure, lowering the soil pH. But all of this, normally you can find studies that have success, but also studies that didn't have success. And sometimes you have year-to-year variability also. So we're looking into approaches that could reduce common scab. And today I'm going to present two that I've worked on. The first one is increasing resistance of known variety using somoclonal variation. And the second one is exploring what factors explain the presence of common scab pathogens in certain zones of field or in a commercial potato field. So the first study is about somoclonal variation. So what is that? Well, that's basically the natural ability of plant to change under stress. And that occurs very frequently in tissue culture. And who knows, like you cut the poor plant in bits and put it into artificial medium, and that's stressful. And actually, this is working for us because we can find things that are improved. And so that was the objective of this study, was to increase resistance of known varieties. So we picked Chepedy and Red Pontiac because the industry wasn't interested in those two varieties. So just to give you a very brief overall overview of how we do this, the first thing one needs to do is to find grow conditions where you could see a multiplication of the cells. And it's not every medium that will allow this. So if you look at, like if you look in week 6, you kind of start to see that the cells are multiplying. It makes kind of almost like a chewing gum look. So that means they're multiplying. And what you want is basically to see plants regenerating. And at week 12, you can see that basically the plants have succeeded in regenerating. So that's success. The second step was to look at using actually the plant toxin, the taxtamine that is produced by all pathogenic Streptomyces species, and to use that as a way to kill any cells that were susceptible. So we're increasing the number of plantlets that would be resistant to common scab. So that's what we did. We tried different concentration of taxtamine, and you can see at the low taxtamine concentration, they kind of start to be a funny green. And when you put a high concentration of taxtamine, you can see that they're struggling and they're becoming quite brown. but they're still regenerating some plants, so that they're quite difficult to kill, those potatoes. So what we did then, we had 50 variants that we have produced from Red Pontiac and Shipity. We produced mini tubers in the greenhouse, and then we did field trials in 2017 and 2018. So this is the results we got for Red Pontiac in 2017. On the x-axis, you have all the different number for the variants. And on the y-axis, you have the percentage of lesions covered. So how much lesion do we see on the tubers? And that is indicative of how severe the common scab was. So the first one is named PL. That's the parental line. And that's what we're comparing with because that one is kind of our control. So we see if the variants are worse or they're better. And what you can see that some of the variants are actually worse than the parent. So basically, the severity of the disease was greater than for the parental line. But for some of those variants, and they're the blue bars, actually have a nice decrease of 2 to 3 times less than the parental line in terms of severity of the common scab. So basically, for Red Pontiac, we've chosen 12 variants that were better. I'm not showing you the Shepherdy, but it's the same type of trend we have noticed. Some were worse, some were better. We actually have better than the parental line. Now this is now an in-depth evaluation, and I'm showing you Red Pontiac and Shepherdy in 2018, but the results were very similar in 2017. So you can see here again that most of the variants are performing better than the parental line, and some of them like 25, 44, and 48 for Red Pontiac, 64, 100, 106, and 110 were variants that were consistently better than the parental line, both in 2017 and 2018. So now we were interested to see, well, it's nice, but what does that mean in terms of how much more do a grower gain by using this variant? Would you get more? Would you make more money, basically? And so what we did is we basically look at the number of tubers, basically that have now less than 5% of lesion coverage. And in the industry, there is a rule that if you have less than 5%, then it's sellable. You can sell your product to the table market, and you don't get hit also when you go for the industry. And so what you look here is what you want is your variant actually have more potatoes. So now you want the bars of the variants to be better than the parental line. And what we see is actually most of the variants have like a significantly more tubers that have less than 5% lesion coverage. So that's excellent. You can see that the variant 25, 44, and 48, the best one for Red Pontiac are very good. And for Shepherdy, again, we also have like very good results where we have a lot more tubers now that would be sellable for the growth. So that's excellent. And the results for 2017 and 18 are very similar. Now, we also looked at the relative marketable yield because, like if that would be bad if your variant was suddenly producing not as many tubers. It might be cleaner, but you're still losing. So we checked that last year. Unfortunately, last year, here in Fredericton, it was extremely dry. So it might have not been the best year to actually look at the marketable yield, but anyway, you have to start somewhere. So for Red Pontiac, now what I did is to make the relative marketable yield. So this is the variant compared to the parental line. So if it's perfect, it would be 100%. So now you want basically the variants that are above the line, above 100, means that they are actually having more yield. They're producing more tubers than the parental line. But when they're below, means they're actually not performing as well as the parental line. And so what you see for Red Pontiac is that actually the variant 25 is not performing very well. So it might have been one of our best for severity, decreasing of the severity of common scab. But it seems to be, at least for this year, having some reduction in the marketable yield. But this is not the case for 44 and 48. So we'll have to monitor the situation. Shepherdy seems to be struggling a bit more, but it actually was very susceptible to drought. So I'm not sure we can believe the results, but there is certainly some reduction in the yield. So we'll have to monitor this closely in the future. So in conclusion, somoclonal variation was successful in finding variants that were better than the parental line. And there was about a decrease of 2 to 3 times in terms of the severity of the disease. And for those variants, there was about 20 to 40% increase in terms of the number of tubers that have less than 5% in lesion coverage, so more that the grower could sell. But there's really need to have more evaluation on this, just to see if the trend in resistance will also be seen in multiple sites, and if also continue to monitor if there is actually a hit in the marketable yield from those variants. But it seems to be a possibility in terms of for growers to lose somoclonal variation to increase comet scab resistance. Okay, so similarly to George, I also have lots of questions about why is it that my fields, this magnificent fields, everything is good except in that area, I have a lot of comet scab and I'm having a problem because then when I plant there, I lose a lot of my material. And so we know that the distribution of pathogens is always highly variable, but there are some regions and some fields that are really performing poorly in terms there seems to be a lot of the pathogens and very abundant. And the question was, is it about soil properties, or is it about soil microbial communities, or is it about both? And so what we did, we actually selected 9 sites that were actually paired fields, so one infested versus one control. They were located in New Brunswick, in Quebec, in PEI. We went 3 times over the summer, in May, in July, and in September. We took the bulk soil and we took the rhizosphere. And in all of those fields, we actually planted red Pontiac. And so we had 3 replicates. So if you see at the box at the bottom, it kind of replicates potato rows. And then we would go and remove the tubers and put the red Pontiac in the healthy fields, in the infested fields or control fields. And so what we did after is we did all kinds of measurements. So to measure the soil physical chemical property, I'm not going to name them all, but they're, we basically did all we could think of. And then we also did biological measurements. We looked at the disease severity of common scab at the end of the year. We quantified the pathogens by quantitative PCR, and we also looked at the bacterial and fungal community using next generation sequencing. So in terms of disease severity across the sites, you can see here that in the x-axis you have the sites. In the y-axis you have again the severity of disease in percentage. And what you can see, the red bar are the common scab infested, and the blue bar are the control. So in most cases we have a very important difference between the infested zone and the control zone. And you can see on the right some pictures to show you when the infested PEI tubers, they had no skin left, and they are actually difficult to find in soil because they were all brown. So that one was our extreme case. Now, in terms of the results, here, it's a different way to visualize correlation. And so what you see here is that when it's red, it's a positive correlation. When it's blue, it's a negative correlation. And the size of the circle indicate how strong the correlation are. And so we were particularly interested in seeing what was correlating with disease severity. And I put into the corner in the box what was correlating very well with disease severity. And we have pH, which was not a surprise. We also have potassium, as similar to George. We have phosphor, calcium, magnesium, iron. Carbon was also very well correlated with disease severity, and the CN ratio. And so that's basically what we found for this. Now we also look for what was the difference in the diversity of bacteria and fungi between control and infested field. So the first graph that you see at the top is actually the bacteria. The one on the bottom is about the diversity of fungal diversity. And what you can see is that there are differences between the control and the infested, and so that's indicated with the stars. But not all fields, there was really a significant difference between bacteria and fungal, between healthy and infested. Now, we also look at correlation between the diversity of bacteria and fungal communities and the severity of disease. Again, the red is a very positive correlation, blue is negative. what you can see is like community is actually very much in the positive correlation. But what was interesting, and if you look at the disease severity, the green box is all about bacteria at the 3 different dates, May, July, and September, and they're correlating with disease severity much more than the fungal communities. And so we think that indicates that if there is a role of the communities in, differentiating those infested fields, it's more about the bacteria than it's about the fungi community. So what's important, what's different between infested and controlled fields? Like, why is it that some fields are clean and some are not? Is it about soil property? Yes, there seems to be important differences in the soil physical chemical property, particularly the pH, the potassium, the magnesium, iron, and phosphor. But it's difficult to know, especially about the pH, because the pH, we may not sure if it's a direct effect or an indirect effect, because the pH has such a very important role in solubility of those ions that it might be actually an effect of the pH we're seeing. But we definitely need to have more research on that. So was there differences in the biological factors between infested and control zone in the fields? Yes, there were differences, but not always. But there definitely has some, and the diversity of bacteria was correlated with common scab severity, but not the fungal communities. And so the results suggest that both the soil physical chemical property and the microbial communities are associated with common scab severity. However, we will need more study to really see if this is really the cause of differences between fields. So the overall conclusions is that for common scab, there's unfortunately no silver bullet. There are some practices to control common scab, like biopesticide, reducing soil pH, application of sulfur or manure, that may work. But the response very much depends on the soil property, the environmental conditions. And so if you want to try that, you really need to test it under your condition and evaluate if that works. And normally you want to test that on a small zone of your field, not everywhere. Because if it's not working for you, then you don't want your field to become worse than it is. And basically, the only thing that really, really work well is using resistant varieties. And that's the best strategies, because normally some of them are really good. You have to test that under your conditions, but it normally is the best approach still. Unfortunately, I know some of you are really attached to your variety, but sometimes if you have a zone that is very difficult to and you don't know what to do, resistant variety is something you should really consider. And that's it for me. Oh, no, that's not true. I just wanted to mention briefly, we have a new project under the potato cluster that was funded by the CAP, the Canadian Agricultural Partnership, and with the CHC being the applicant, where we are going to continue and looking into trying to better understand what the mechanisms are when we're controlling common scab. Is it about soil property and environmental conditions? And to do that, we'll have a mix of different agricultural practices that we want to try, like nurse crop, biopesticide, compost, mustard meal, etc. And we also have another component where we're also looking at genetic diversity of pathogenic streptomyces in New Brunswick, PEI, and Manitoba. And we really want to push the analysis to really look at the genome of those pathogenic the pathogens to really see how much differences there are genetically. So I think now this is the end. Thank you.
Thanks so much, Claudia. In the next segment, our guest is Dr. Yahoo Wang. Yahoo is a professor and potato researcher at Dalhousie University, the Faculty of Agriculture in Truro, Nova Scotia. The floor is all yours, Yahoo.
Hello, everyone. I would like to thank SpudSmart for giving me the opportunity to share our study with you today. And the topic I'm going to talk with you is biocides used to control common scab in potatoes. And I would like to share with you the results from our 2-year field trials. First, I would like to acknowledge the people who have involved in this research. First of all, Dr. Robert Coffin and Joyce Coffin, who offered the field trials and also performed planting, treatments, management of the field and the harvest. And so the whole 2-year field trial was carried out at private farm in PEI. And I also would like to thank people in my lab, which are all listed here, for Scab reading, suberin analysis, statistical analysis, as well as Brian Beaton and Claudia Goyer for developing the project with me. We have heard a lot about common scab from the previous 2 speakers, so I'm not going to get into greater details on them. This is my summary about possible options for control common scab. Number 1 importance is to get resistant varieties if you can. And number 2 is soil fumigation, which is very much limited right now throughout Canadian potato growing areas. And the third one is the usage of biopesticides. And the fourth one is just somoclonal variations with existing varieties of potatoes, which Claudia had mentioned a lot today. So what I'm going to focus on is the use of biopesticides that could or could not be used for common scab. In our 2-year field trials, we have 3 treatments and we chose 3 products. Number 1 is called Microflora Pro, and this is the bacillus-based product that you can get in Canada. And the second product is called Double Nickel 55, which some of you may have heard. And so those are all promising products for us. So we thought we'd give it a try to see how they work. The third product is a fungicide. It's a phosphate fungicide that is not claimed to control common scab. However, more and more potato farmers have been using them to control mostly late blight or silver scurf before putting potatoes into the storage. So we wanted to see if phosphate alone or together with other biocide could affect the common scab. And we did in-furrow applications of microfurrow and double nickel, and with recommended rate, which is Microflora Pro 800 mL per acre. Double nickel is 1,000 grams per acre. And for the phosphate fungicide, we chose 1 product, which is Fostrol. And we did 4 times foliar applications during the growing season. And we chose 2 applications at a lower rate first, and then we chose the second 2 applications with a higher rate. So we use 4 varieties: Gold Rush, Green Mountain, Prospect, and Red Pontiac. And we made 6 treatments, and the first 3 would be Check, which has no treatment, and then Microflora and Double Nickel. So those are 3 treatments. And the last 3 treatments are the same as the first 3, but with Fostrol added. So we together have 4 varieties, 4 replications, and 6 treatments. Altogether, we have 96 subplots. And we did, this is the field at Roberts Farm in PEI in 2016. And this is in 2017. Both fields are known to have very severe common scab inoculum, and that's why we are looking at if common scab could be developed in those 2 fields. We did harvest in on October 13th in 2016. So altogether we have 96 bags, each for 1 subplot, and we took about 25 to 30 tubers per plot. And our harvest date in 2017 was October the 5th. Again, we took 96 bags, around 30 tubers per bag. We brought them back to Truro, where I'm located, and we did all the lab analysis. First of all, Zahid, my technician, was washing every tuber carefully. Every bag has 30 of them. And then we photographed all the tubers on both sides. So the side one, we have them photographed, and then we turn them around and we photographed side two. Oops, sorry. And we did scab evaluations as well. We did 4 types of scab disease ratings. Number 1, we looked at the percentage of scab area per tuber, every tuber. And second, we did number of shallow pits per tuber, which would be less than 4 millimeters in depth. So Zahid has to use a knife to cut the pits to see if they're shallow or they're deep pits. So the third one is we counted the numbers of deep pits per tuber. And then the last one is we measured the necrotic areas per tuber. So as you can see, this is tedious job and very labor intensive, but without this is necessary to categorize the disease in different aspects. In addition, we also did tuber skin analysis. As you can imagine, the reason the scab could be developed is somehow the pathogen had penetrated the skin or distracted the skin structure. So what we did was we looked at the most important component, which is suberin in the skin, and we collected the data on the number of suberized cell layers and the thickness of suberized cell layers. This is microscopy analysis, which I will show you how we did it. So this is the our early publication. So the methodology was established in our lab. We were able to see the skin structure, which is on the B, on the right side of the screen. Under the fluorescent microscope, we were able to stain the skin slices, and we are able to count the numbers of cell layers, as you can see here. In addition, we can also measure the thickness of the cells, because both are important features against common scab. And this is my student, Ying Yi, who were cutting the tuber's skin and do all the analysis and put them on the spreadsheets. We used 8 to 10 tubers per bag for all 96 bags I mentioned before. So now I would like to share with you the results. As you can see, the genetics play a great role in resistance to common scab. Golden Rush obviously is very resistant, and Red Pontiac is very susceptible. And the other 2 varieties, Prospect and Green Mountain, are falling in between. Yes, so I have already talked about this already. Red Pontiac that are treated with double nickel and microflora has shown some visible differences, especially when microflora was used. The skin was brighter. However, after 2 years, we couldn't see severe improvement, which I'm going to show you very soon. This is the double nickel treated Prospect. There's maybe a little bit visible differences you can see with double nickel treatment, but not significant treatment with microflora. And so this is the statistical analysis we performed. So I have many tables to show you. This is one of the first ones. It's disease severity, which means the percentage of tuber surface with scab. As you can see, there are many numbers here, and they also have letters. The ones that shown in green are positive. The ones that are shown in red are negative. For instance, in Gold Rush, double nickel treatment, control and phosphide, and microflora and phosphide are having negative impact. and the others don't have significant impact. In Green Mountain, microflora has negative impact, whereas double nickel plus phosphide has positive impact. In Prospect, double nickel has negative effect, whereas double nickel plus phosphide has positive effect. And so double nickel with phosphide is really helping Green Mountain and Prospect with disease severity. And in Red Pontiac, the only negative impact comes out is control plus phosphide. In regards to shallow pit, as you can see, only one red showed up on Prospect, which is double nickel treatment, which means double nickel will make the shallow pits more severe, whereas the other treatments have positive impact that includes phosphide, double nickel and phosphide for Prospect, and microflora plus phosphide in both Green Mountain and Prospect, which is quite promising. Oops. In regards to deep pit, you can see the positive effect with phosphide in Green Mountain and double nickel plus phosphide in Red Pontiac. And all others either have no effect or negative effect. Necrotic area, the only treatment that makes it worse is double nickel on Prospect. And this is the data from the last table. I just wanted to show you, even we present the data on the figure, how it looks. Okay, so this is a really interesting feature I would like to present to you, which is the skin structure, the superized skin structure in 4 varieties. As you can see, Gold Rush has very well packed, organized, superized cells on the skin, and they're also quite thick. Okay, so they have more layers of skins and also more layers of cells in the skin, and also very thick. When you look at Prospect below on the left, you can see the skin is not well organized, and also the cells are bigger, and the skin is very thin, and the layers of skin is much less. And Green Mountain as well is not as thick as Gold Rush, and it's not as many layers of cells, and the cells are rather big. And the top bottom on the right, the right bottom is Red Pontiac. You also see very similar features, which is much less skin layers, and also the cells are not so well organized. And this only evidence could demonstrate why Gold Rush is a resistant variety and the others are not. Okay, so superized skins in 4 varieties. If you look at the top data, which is the number of superized cell layer, you can see the most is Gold Rush, and then followed by Red Pontiac, which is a B, and then Green Mountain, which is a C, and then Prospect, which is the least, that's a D. And the thickness of the whole superized cell layers, and definitely Gold Rush has the most, followed by Green Mountain and Prospect, and Red Pontiac has the least. And that's why Red Pontiac is also a negative control. Okay, so this is a complicated slide, and actually that's the very last one I'm presenting to you. This is the slide summarizing if any treatment have increased the number of superized cell layers or the thickness of the superized cell layers. If you look at Gold Rush, they're only red, which means double nickel plus phosphide had decreased the number of cell layers, and the all bottom 4 treatments have reduced the thickness of the superized cell layers. This is for Gold Rush. However, when you look at Green Mountain, almost all the treatment have improved the number of superized cell layers and the thickness of the cell layers. And in Prospect, only 2 treatments gave positive effects, and the other treatments did not have any effect at all. Whereas in Red Pontiac, the last 2 treatments, that is double nickel plus phosphide, as well as microflora plus phosphide, have negative impact on the both numbers and the thickness of the cell layers. So as you can see, all those treatments do give different perspective on the outcome of the severity of common scab. So in conclusion, common scab resistance is cultivar dependent. Common scab severity should be carefully evaluated by many parameters, as I have shown you. Also, the effect of biopesticides is cultivar dependent. You cannot say it's working or not. It has to be seen by different varieties. There is a positive or negative interaction between the 2 biocide and phosphide on the severity of the common scab. And the skin superization features are strong indicators of the scab resistance and susceptibility, which could be used for cultivar development or screening in the future. So with that, I would like to acknowledge the funding agencies who have supported this research. Thank you.
Thank you, Gefu, for your presentation. If you have not already done so, please type any questions you may have or comments into the chat box, also indicating to whom the question or comment is directed. The first question, George, I thought maybe you would like to go over this question that appeared in the chat box. Is it likely that the difference in response in Ontario and PEI soils relates to different soil types, such as clay versus sand, as well as different bacterial species?
It's to me?
Yes, please, George.
Well, both the Ulster soil is a silty loam, but it's very sandy in nature, as was the PI soil. So, as far as structural components, they are very much very similar. But there's no doubt that I think the bacterial species present have different virulence components, and we need to find out how each of these species behaves, and we know very little about this.
Now, for each of the guests that we have on the line, I was hoping you could answer, what are your next steps for your particular common scab research, and what do you believe is the key to unlocking effective methods of control? Can we start with you first, George?
Oh, yes. Well, it's not that scab cannot be controlled. In Israel, they have a major scab problem with peanuts, which are a very, very high value product. And for that, they have used a product called that's formaldehyde. It's a formalin product that's registered in Israel. While it was considered as a potentially harmful product, it's been shown that the analysis that was done on it originally was incorrect. And they use it extensively for disease control as a fumigant. But it has... I don't recommend it because it has secondary impacts in that it kills a lot of bacteria. So we tested it in Canada, and what we ended up with is little black golf balls because Rhizoctonia went nuts after killing off all the bacteria. I think seed treatment overall is going to help, and I think eventually we're going to find some kind of microbial management technology that will work. We tried a number of biocontrol agents, but sadly, we only tried it against one species rather than multiple species. And that's what we're going to look at currently, looking at biocontrol with multiple species of bacteria, or there's multiple pathogen species of Streptomyces.
Thank you, George. Claudia, do you have any comments on next steps for research or what you believe is going to unlock some really good effective methods for common scab control?
Yeah, I guess sometimes I'm reminded, like a common scab has been discovered like in 1898, right? Like, and we're still struggling with that disease. So it is a very complex disease. Part of the complexity, I think also is that it's a bacteria. So for fungi, soil-borne disease, we have fungicides. But for bacteria, you would need antibiotics, and we can't because we already have lots of problems with highly resistant bacteria in the hospital. So we don't want to compound that problem even more. But that leaves us with a very few kind of chemical approach that we can reuse. And so I think we're still, I think there's still some value in looking again at some of the agricultural practices. I think we have new tools to look at the effect on the pathogens and the effect on the communities and looking really at the soil property. I mean, the project I was talking about in the cluster, we're really trying to understand why is there so much variability site to site. And we're hoping that once we understand when it's working and when it doesn't, we're better equipped to apply the agricultural practices in the right way and in the right place too.
Thank you, Claudia. And Geffa, do you have anything to add to that?
Yes, I have studied many diseases in potatoes, such as late blight, verticillium wilt, fusarium, and common scab. And also I have a project on wireworm. So my interest for the next step is look at the microbial community in the soil, focusing on soil property and how the soil features are interacting with the microorganisms. I don't know what I'm going to find out, but I think all the pathogens interact with each other, causing different diseases. And it's really difficult approach, but I think that's something that is really important for us to look at.
Thanks so much, Gefu. Well, that appears to be all the time we have today for questions. I would at this time like to thank our guest speakers for sharing their insights and expertise during this SpudSmart webinar. I would also like to thank our sponsors today, BASF and McCain Foods, for their support. This webinar will be available on SpudSmart.com if you'd like to view it again. And on behalf of the team at SpudSmart, I'd like to say thank you for all of you for your participation, and I hope you go on to have a fabulous day. Thanks so much, everybody, and we'll sign off now.